The Process of Photosynthesis by
Kenzie Doherty,
Emily Colby,
Emily Felton,
and Charlie Hangspaw.
The general equation for photosynthesis is CO2 plus H2O plus light energy yields C6H12O6 plus O2. This is a biochemical pathway, meaning that the inputs of one are the outputs of the other.
Requirements for photosynthesis are water, carbon dioxide, sunlight, NAD, pH, and ATP.
Photosynthesis consists of two major parts, the light reaction and the Calvin cycle.
Water in the light reaction splits into hydrogen, oxygen, and electrons. Carbon dioxide in the Calvin cycle combines with RUBP. Sunlight in the light reaction gives energy to pigments, chlorophyll A, chlorophyll B, and carotenoids. NADPH is created in the light reaction, but gives a hydrogen and takes a phosphate from G3Ps in the Calvin cycle. ATP is created in the light reaction and gives a phosphate to PGAs in the Calvin cycle.
Like previously stated, thylakoids are in the chloroplast and are where the light reactions take place. The thylakoid membrane consists of a phospholipid bilayer. Photosystem II and photosystem I are integral proteins that contain pigments. These pigments are chlorophyll A, which is in the reaction center and absorbs more red and less blue wavelengths. Chlorophyll B and carotenoids are outside the reaction center and capture what chlorophyll A doesn't. They absorb more blue and red wavelengths. NADP plus reductase is also an integral protein that creates NADPH. ATP synthase is another integral protein that creates ATP. Electrons travel along the electron transport chain between photosystem II and photosystem I. The short ETC transports electrons between photosystem I and NADP plus reductase. The lumen is the space between the thylakoid membrane.
Water breaks down into three things: hydrogen, oxygen, and electrons. The oxygen is released through the stomata as a waste product. Hydrogen is pulled into the lumen and is also used to create ATP through the process of chemiosmosis. The electrons go to photosystem II. NADPH is created from electrons going through the NADP plus reductase and combining with NADP plus. Chemiosmosis is used to make ATP. This occurs when the hydrogen funnels through the ATP synthase and gives it energy so that the ADP can combine with phosphate to make ATP.
These products, made in the light reaction, then travel to the Calvin cycle. The Calvin cycle takes place in the stroma.
The Calvin cycle begins with carbon dioxide combining with RUBP through the rubiscal enzyme. This becomes a short-lived 6-carbon compound, which immediately splits into PGA.
ATP comes in and adds a phosphate to the PGA, making them biphosphated PGAs. ATP exits the Calvin cycle as ADP and goes to the light reactions. NADPH then comes in to remove a phosphate and add a hydrogen to the biphosphated PGA, making them G3P, also known as PGALs. NADPH exits the Calvin cycle as NADP+, and returns to the light reactions.
One G3P exits the Calvin cycle to create half of a carbohydrate. This could also produce proteins or lipids, not only carbohydrates. The rest of the 5 G3P continue in the Calvin cycle and with the help of ATP become the 5-carbon compound RUBP once again. The environmental influences on photosynthesis are temperature, light intensity, and carbon dioxide concentration.
As the amount of carbon dioxide increases, so does the rate of photosynthesis, but only up to a certain point. At a certain point of carbon dioxide concentration, the rate of photosynthesis plateaus.
As the amount of light intensity increases, the rate of photosynthesis increases as well. At a certain amount of light, the rate of photosynthesis plateaus.
As temperature increases, so does the rate of photosynthesis, but only up to a certain point. At a certain temperature, the rate of photosynthesis decreases because the integral proteins are unable to work.